First of all, methylchlorophosphate derivatization is an ideal derivatization technique for amino acids. It yields the respective esters and carbamides. And for this class of derivatives, we first noticed that there's an influence of differences in humidity in the lab with regards to the ionization. And therefore, or in fact, we observed that the protonated molecular ion was no longer more the dominant ion in the mass spectrum, but instead a fragment ion was the dominant one, which can be a problem because in metabolic fingerprinting, if you want to identify unknowns, you would not know from which mass to start for your identification. And therefore, we try to continuously infuse water from the top of the APCI source to provide a constant level of water traces which are required for the ionization, and that improved the situation a lot. And for all the compounds from our test mixture, we obtained the protonated molecular ion now as the base peak of the spectrum, and well, this improves the situation in metabolic fingerprinting if you want to do identification. And we also saw this in a metabolic fingerprinting approach when we compared cancer cells that were treated with an HSP90 inhibitor, 17-DMARC, compared to control cells. We saw that in PCA plots, the clusters of the biological groups were much tighter with water infusion, so variability distinctly was decreased. And this was also seen when we looked at the identified significantly regulated metabolites between the groups. With water infusion, we could increase the number of compounds detected and identified from 13 compounds to 23. So about 2 times higher was the number of identified metabolites. And this is here in metabolic fingerprinting approach proven, but can also, water infusion can also enhance the performance in a targeted analysis, what we did not do in this study here, but there are papers out where the analysis of pesticides is improved by water infusion, and it leads to more abundant and more selective protonated molecular ions for these compounds.